Hello students, today we are going to discuss a very important topic which you have heard already, I believe, and that is DNA fingerprinting. Now, if a person have, for example, lost or he or she had been involved in a criminal activities, this is one of the methods by which the forensic sciences they use to identify the person who have been involved in different activities. So, if these techniques, is used in forensic sciences, it means that there is some reason behind it. That is the theoretical or the precise application. If a person have done something wrong, it means how precisely these techniques can find out the person behind the activities. So such techniques can also be used, for example, in the genetic populations, to search the populations of different individuals. And not only that, like genetic disorder diseases can also be detected from these diseases, like from the parental to the offspring. Those genetic diseases can also be detected. It was Alec Jeffrey who have developed this technique. And before going on to the process, let's discuss some of the important requirements of this process. The first is your VNTR, and which is also said to be satellite DNA or mini satellites. Mini satellites. So VNTR means variable number of tandem repeats. I'll write here, variable number of tandem repeats. Usually its size used to be 0.1 to 20 kb. Kb means kilo basis. Why this is very important? Because it shows high degree of polymorphism. Now, what is polymorphism? Polymorphism means variation in genetic level. Variation in genetic level. Now, this variation in genetic level is because due to the mutation, right? So, since this satellite DNA they provide high degree of polymorphism, they can be used as a probe. It means that VNTR is used as a probe. So when I say probe, remember that it's a VNTR, that is variable number of tandem repeats. Now to perform this technique, we need DNA as a starting material, right? So the first step is isolation of DNA. So from the human cells, DNA have to be isolated, and for that we use chemical process or the enzymatic process. We use some degrading enzymes to break the cell wall, and then or the plasma membrane, and then we take the DNA. We isolate the DNA. After isolation of DNA, it has to cut with restriction enzyme. Now, as I said, restriction enzymes are also said as, they act like a scissor, and depending on the requirement, they are very specific. They can cut at any specific point depending on our desired fragments. So they will cut the DNA into fragments, and we call as molecular scissor. Molecular scissor. Regarding this restriction enzyme, we are going to discuss in detail in biotechnology, that is chapter number 10. Now, this enzyme will cut DNA into fragments. After that, these fragments have to be separated. Just say that this is our test tube, okay? And then in this test tube, you have added restriction enzyme, and this was the DNA, okay? This restriction enzyme will cut and it will form many fragments, okay? Once the fragments has been formed, they have to be separated. So this is one technique, separated by gel electrophoresis. Electrophoresis. The fragments, that is, just say that this is DNA fragments. Now these fragments are of different size, okay? Different size. Some will be short fragments, some will be long fragments, okay? So there will be long and short fragments. It means that with the help of gel electrophoresis, just say that this is a machine, okay? Actually it's a machine only, and then you're going to put this. I'm not going to discuss in detail about this gel electrophoresis, but these DNA fragments will be added in this machine and they will run. After some time, you will find that these DNA fragments, you know that DNA is negatively charged, right? So when a current is passed, they will move, okay? And the smaller size, okay, the smaller size will be the, see, can you see that this fragment is in the living away, okay, going away from this well, okay? So this is said to be the small fragments, and the one which is nearby to the well, okay, just say that this fragment is nearby to the well, okay? So you can say that this is large fragment. We're talking about DNA, okay? So it means that depending on the size, we have separated the DNA fragments. Once it is separated, we take out that DNA fragments, okay, depending on our requirement, okay, and then we take out that DNA fragments and put in a nitrocellulose membrane. It's like paper, okay, which is made up of nitrocellulose, okay. We'll transfer these fragments in this membrane. Then we will label it with a probe. So what was the probe? The probe was VNTR. Yes, sir. Variable number of tendon repeats were used, okay. This probe was hybridized. Hybridized means if there is a in that DNA fragments, if there is a AT, G and C, okay, it means that our variable number of tendon repeats will be T, A, C, G, the complementary strength, okay, and it is radio labeled. Now after that, they will observe in a radiography. So when they observe in autoradiography, they will see, we can able to see the different bands. Now if I take my chromosome, okay, and perform a DNA fingerprinting, obviously I will observe different bands because I have different sequences of DNA. Now if I take your DNA and then proceed the same steps and then perform the DNA fingerprinting, then if I see the band, it will be a different band because your sequence is different, right? So in that way, okay, we can able to differentiate the different sequences of the DNA for different individuals. So this application, okay, the first step is the application of this DNA fingerprinting, okay, is as I said, for the forensic sciences, in clinical, okay, in the field of diagnosing the diseases, okay, and even to know about the genetic population of any species. So the first step is isolation of DNA. The second is we have to cut the DNA fragments with the help of restriction enzyme. Okay, restriction enzyme. Third, we have to separated DNA fragments by gel electrophoresis. After separation, we have to transfer to the nitrocellulose membrane where we have to hybridize. Okay, hybridize. After hybridization, we have to observe at the autoradiography. So this ends the chapter of molecular basis of inheritance. I believe that the video which I have prepared for you, I want all of you to observe, listen, and if there is listen properly, and then if there is any problem to be discussed, okay, if you have not understand the process properly, you can call me or you can give a message in the WhatsApp so that we can go for the discussion for proper understanding for the concept. Thank you.